Thank you, Chairman. I am Daisuke KATO from the Department of Radiology at Hokkaido University Hospital, Japan. The title of my presentation is Functional Analysis of AQP4 using Wara and Solute tracer in MRI. I have no conflict of interest to disclose. Since the green fatigue cyst hypothesis was proposed by Dr. Arif in 2015, there has been more awareness about the importance of cerebrospinal fluid (CSF) in the lymphatic function of the brain. This concept proposes that CSF enters brain through the tiny spaces known as perivascular spaces, located in the arteries on the brain surface. Here, it circulates through the brain parenchyma and eventually leaves the brain through the space in the veins. This circulation of neural fluid is considered to contribute to the movement of substances within the brain, and it also helps in the removal of waste from the brain to the extracranial space. A key protein in this process is aquaporin-4 AQP4 water channel, found particularly in the end feet of astrocytes around the small vessels. AQP has been suggested to facilitate the flow of interstitial fluids CSF-ISF exchange, and it is considered to play an important role in the green fatigue system. One method for examining comprehensive CSF movement and green fatigue system employs intracerebral tracer-injected MRI. First, the intracerebrally injected tracer spreads from the caudal to the dorsal CSF spaces, and it is then transferred into the brain parenchyma. From there, it is drained extracranially through several pathways. This approach effectively displays the flow and dynamics of neural fluids. However, it might not truly represent neural fluid dynamics given the distinct behaviors of solid and water molecules. As such, we advocate for the use of water tracer as well. We have placed a particular emphasis on O17 level water owing to its unique advantage. O17 level water allows us to track water dynamics over an extended period using MRI. Additionally, there is no radiation exposure, and it can be administered harmlessly. Given these attributes, we see O17 level water as an optimal choice for assessing AQP4 water channel. Note that O17 water MRI has some challenges, and its application is not easy. Hence, we'd like to initially start with intercerebral gadolinium MRI and then advance to O17 MRI. Several reports have been published using intercerebral gadolinium MRI in various pathologies. For example, this report of intercerebral gadolinium MRI in a tauopathy mouse models shows that tracer transfer to the brain parenchyma was reduced in the disease model. As such, the inhibition of the green fatigue system is depicted as a reduction in tracer translocation into the brain parenchyma and washout. However, there are limited reports on the functional imaging analysis of AQP4, and there are uncertainties and a lack of consensus. Therefore, we initially performed the functional imaging of AQP4 using intracerebral gadolinium MRI in AQP4 knockout rat. This slide shows an outline of an intracerebral gadolinium MRI experiment. 3D T1 weighted images were acquired over time during a 30 minutes intracerebral administration of gadolinium tracer to each of the knockout and wild-type rats. Some parameters of MRI are shown in the slide. After post-processing of the image by motion correction and anatomical standardization, a signal ratio map was generated to visually assess the difference between the two groups. Several ROIs were placed for CSF-ISF exchange and CSF drainage pathway and CSF spaces. Thereafter, the time intensity curves were plotted and compared. AQP4 knockout rats established by Dr. Yasui at Keio University were provided. He's an expert for AQP4 and the last speaker for Trey. Thank you, Dr. Yasui. This is the result of the CSF-ISF exchange analysis. Although the difference between knockout and wild type are not obvious in the signal ratio map, they become significant in the subtraction images of both. In the subtraction images, colder color means that the knockout group has a lower signal relative to that in the wild type, and warm color means the opposite. Knockout group had a lower signal in the brain parenchyma than their wild-type rats, such as olfactory bulbs, ventral and lateral cerebral cortices, and hippocampus. This suggests that tracer location into the brain parenchyma was reduced. In the time intensity curve plots, the blue and red lines represent the wild type and knockout model, respectively. In some rows, such as the olfactory bulb, cortex, and hippocampus, the knockout group showed significantly lower translocation of the intracerebral tracer into the brain parenchyma. This result suggests the loss of AQP4 reduces a CSF-ISF exchange. Next, we present the result for the CSF rags. Similar to the previous observation, the subtraction images of the two groups showed significant difference. In CSF drainage pathways such as perioptic nerve cells, nasal cavity, and dry lymphatics, the color of subtraction image was colder. This means that the intracerebral tracer translocation was reduced in the knockout group compared to that in the wild type. In contrast, the color of the subtraction images in the CSF space, frontal, dorsal, ventral, was warmer. This might indicate that the tracer in the knockout group is stagnant in the CSF space. The time intensity curve analysis also shows similar results. The knockout group exhibits a reduced tracer translocation into the CSF drainage pathway compared to that shown by wild type. And consequently, the tracer accumulation in the CSF space was observed in knockout group. We speculate that AQP4 deficiency may impair CSF solute clearance and lead to CSF stagnation. This slide summarizes the result. Intracerebral tracer MRI shows tracer translocation from the CSF to the brain parenchyma, which then drain extracranially through several pathways. If the AQP4 function are lost, the tracer translocation to the brain parenchyma is decreased, suggesting reduced CSF-ISF exchange. Moreover, the translocation of the tracer to the drainage pathway decreases, and tracer accumulation in the CSF space increases. This might mean that CSS solid drainage was restricted and led to CSS stagnation. These results are almost compatible with previous reports where AQP4 inhibitors or other species of knockout models. However, some difference was observed. For example, smaller changes in AQP4 deficiency relative to control were observed in our data compared to previous reports. We consider that this might be related to the acute or chronic nature of the loss of AQP4 function, was difference in experimental techniques such as tracer injection parameters or anesthesia. These results represent the solute tracer behavior, so water tracer research is needed to investigate the CSF dynamics. Based on these results, we intend to perform functional imaging of AQP4 using intercecal O17 level water MRI in AQP4 knockout rats. Based on our experience with O17 MRI, we experimented with O17 water as an intercecal CSF tracer. This is an O17 concentration map calculated from the degree of signal reduction using serial T-weighted images, while intercecal O17 water was administered. As seen, the gradual distribution of O17 leveled water into the brain parenchyma was observed, and we are confident that this approach can be used to assess the movement of water from the CSF to the brain parenchyma. This is a part of preliminary results of intercecal O17 water MRI in AQP4 knockout rats compared to that in wild-type rats. After intercecal injection of O17 water and calculation of O17 concentrations in the cerebral cortex and ventricles, AQP4 knockout rats had slightly reduced transfer of intracecal O17 water to the cerebral cortex and ventricles. The findings from a similar study by another research group are presented in the extracts. They employed an AQP4 inhibitor to temporarily disrupt AQP4 function, and they observed that when using intercecal O17 MRI, the transposition of O17 water from the CSF space to the brain parenchyma became less effective. Our results were similar to those in the previous reports. Loss of AQP4 function also reduces CSF-ISF exchange as a water tracer movement. As shown in the previous study with gadolinium, also in the O17 leveled water study, our results based on the knockout model had smaller effects of AQP4 deficiency than those reported based on AQP4 inhibitors. This might be related to the acute or chronic nature of the loss of AQP4 function. This implies that intercecal O17 water MRI might have potential applications in AQP4 functional imaging. In conclusion, intercecal contrast enhanced MRI with gadolinium and O17 leveled water can be used to visualize solute and solvent kinetics and AQP4 function in neurofluid dynamics. Several neurological diseases have been reported to be used in revealing the importance of AQP4 function. Hence, we'd like to apply this technique to investigate the diagnosis and pathophysiology of this disease. Thank you for your attention.